The term freshwater plants is challenging to define because it encompasses all plants that grow along gradients from land to complete submergence, a distinction that is blurred by variations in water level. Freshwater plants include green macroalgae, charophytes, as well as hornworts, liverworts, mosses, lycopods, and ferns, and also flowering plants that rely on standing or flowing water to complete their life cycle. The different life forms include emergent, amphibious, free-floating, and submerged plants. All freshwater plants, apart from the charophytes, have evolved from terrestrial ancestors. And they include around 5,000 species spread over more than 800 genera and 200 families. The leaves of freshwater plants include the smallest and the largest of all plant leaves. Many contain large volumes of air spaces that produce buoyancy and that allow inorganic carbon recycling, as well as transfers of oxygen from the atmosphere to roots, and gases such as methane from sediments to the atmosphere via roots and leaves. Many water plants reproduce by fragmentation, while some perennial species produce vegetative buds called turions, which function as overwintering organs and propagules of dispersal. Light availability usually determines the depth limit for submerged plants in the littoral zone. Plants acclimate to light availability by altered physiology, leaf anatomy, and shoot morphology. Growth and productivity change through time. In temperate regions, freshwater plants initiate growth once water temperatures reach about 10 degrees. They attain peak biomass in late summer and are killed when temperatures fall below 3 degrees. Plant dry weight is composed of 35 to 50% carbon, but carbon supply can be restricted by low rates of diffusion, which means that high CO2 concentrations are required to saturate photosynthesis. Freshwater plants meet these high carbon needs by growing in habitats where carbon dioxide is high, by exploiting CO2 in the air via aerial leaves or the sediment via lacunae that extend from roots to shoots, and by active CO2 concentrating mechanisms using bicarbonate as the inorganic carbon source. Freshwater plants are integral to the structure and function of littoral zones and shallow lakes. They produce structural complexity and provide refuges and surfaces for colonization. They alter physical and chemical conditions in the water and sediment, and they supply energy and nutrients to the food web. Importantly, several freshwater plants are cultivated for human use. And of these, rice is the most important species, and a food staple for more than half of the world's population. Because of their high productivity, broad ecological tolerances, and easily dispersed propagules, several freshwater plant species are now considered the world's worst invasive weeds, with some species such as Icornia smothering lakes, and others choking rivers and drainage systems. Control of aquatic invasive weeds is costly. It may involve harvesting, introduction of biological control agents, use of herbicides, and habitat manipulation. Dr. Clive Howard-Williams is a specialist in water plant ecology, and he has conducted studies on lake and wetland ecosystems in many parts of the world. Much of his research has been on lakes in New Zealand.
In our deep, clear lakes, such as here in Lake Wanaka, native species of water plants can grow to great depths. Caracean meadows often extend to 30 meters. And in some lakes, beds of aquatic bryophytes are found at depths of 70 meters. Swimming over the littoral zone in our lakes reveals lush and diverse macrophyte communities. But these native plant communities are under considerable threat from submerged invasive aquatic weeds. Most of these invasive species are rooted in the sediments and can extend up to 5 meters through the water column. Which led us to ask the question, how important are roots versus shoots for nutrient uptake? In some of our early work on this question, we conducted a transplant experiment where we took oxygen weeds from oligotrophic Lake Taupo, as shown here, and planted them back in the lake, but using two types of sediments. Sediments from the same lake, oligotrophic Lake Taupo, and sediments from nearby eutrophic Lake Rotorua.
The plants grown in eutrophic sediments grew much faster than in the oligotrophic sediments. These results highlighted the importance of roots for nutrient uptake by water plants, and the capacity for these plants to proliferate through their access to sediment nutrients. This also means that water plants can mobilize and release sediment nutrients back into the water column when they die and decompose. Freshwater plants are directly affected by a changing climate. For example, by increases in temperature, changes in precipitation, and shifts in the hydrological cycle. Evidence from multiple sources, including long-term records, comparisons among regions, and bioclimatic models, point to changes in the abundance, composition, and distribution of freshwater plant communities in response to climate change. The restoration of freshwater vegetation has focused on reversing eutrophication, especially by reductions in external nutrient inputs. Although these efforts can be successful, recovery can take many years to achieve. In this shallow lake in the Netherlands, the charophytes rapidly disappeared with an increase in phosphorus loading in the late 1960s and 1970s. With the subsequent reduction in phosphorus loading, there was a delayed response and the eventual return of charophytes in the 1990s. The many threats to freshwater plants, including climate change, habitat degradation, and chemical pollutants, are leading to the loss of native freshwater plant communities. Many freshwater and wetland plant species are becoming extinct, vulnerable, or endangered. The loss of these plants affects ecosystem processes, functions, and biodiversity, and the ecosystem services that they provide. This highlights the urgency and importance of aquatic conservation measures.